All right, so how many of you did the reading? Cool. You know that you read a little bit about receptors, which seemed like it was from a different chapter, because it was. We're going to talk about an aspect of that, and then another aspect of that we're going to come back around to next time. So what we're going to be doing today is talking about primarily cardiac output, but one of the ways that we affect cardiac output is through changes to contractility, that is how strong the heart contracts. And another thing that we do is we alter the heart rate, which has to do with the SA node cells, right, and how quickly they undergo their action potentials. And both of these things, contractility and heart rate, come down to our body affecting G protein coupled receptors. So that's why this is a part of your reading from that. So before we jump into that alteration of cellular activity, I want to talk through broad concepts about cardiac output, so we're all on the same page. Anybody?
Heart rate times stroke volume.
Heart rate times stroke volume. Okay. Heart rate we define as beats per minute, right? We could say this is a function of the nodal cells producing action potentials per minute, right? So, stroke volume we talked about last time, right? Mathematically, what is the stroke volume? What minus what?
PV minus
N diastolic volume minus N systolic volume, right? And just conceptually, it's the amount of volume ejected from the ventricles. Okay. So overall, cardiac output tells us how much volume is pumped out of the heart over time. Okay. If we have a faster heart rate or a faster stroke volume, either one of these, greater stroke volume. If either of these are elevated, we have an elevated cardiac output. Okay. There's another concept I want you to be familiar with. We don't talk much about both sides of the heart at the same time, but maybe we should do more of that. What is the relationship? We've learned already that the right side has much lower pressure than the left side. Okay. One side pumps the lungs, one side pumps to the entire rest of the body. If we had a cardiac output of, let's say, 5 liters per minute leaving the left ventricle, what would happen on the right side? Would you expect more or less? Would you expect the right and left ventricles to have similar amounts of cardiac output? Is one greater than the other? What do you think? What do you know? They're the same. Okay, they're actually the same. And the left ventricle, even though it's pumping blood at a higher pressure, it's pumping against a greater resistance, right? So we have this high pressure. And then on the right side, we've got a low pressure, and we're pumping against less resistance. But each ventricle is pumping the same amount of blood into either the aorta or the pulmonary trunk. Okay. On the left side, it's got a much longer path, much longer way to go before it comes around again. But in terms of the amount of volume that leaves the heart, they are the same through both sides. Okay. And the last concept I want to jump into is this idea of venous return. I don't think your book defines this, although it spoke about it in conceptual terms at least. The venous return is a value which is essentially the amount of blood coming into the heart per minute. Okay, so cardiac output is the amount of blood leaving the heart per minute, and the venous return is how much blood in the veins returns to the heart per minute. So, how would you expect these values to compare? Yeah?
As venous return increases, cardiac output increases.
Right, so as venous return goes up, cardiac output's going to go up as well. Right? In terms of their value, which one is bigger? Right, neither one. Okay? And I hope this makes sense that the amount of blood that comes into the heart has to equal the amount of blood that leaves the heart. right? This is true on a cardiac cycle basis, right? For every cardiac cycle, the stroke volume is the amount that leaves the heart. That same amount of volume returns to take its place. Okay? And over time, cardiac output is going to be the same as the venous return. Okay, does that make sense? If we understand our two sides of the heart to be interconnected, right? Here they're sort of disentangled, but this is the path. We have to understand that you can't put blood into, let's say box. You can't put blood into this side of the heart of one volume and have a different volume come out, right? You shouldn't. Okay. Over time, you're gonna have a problem, right? So those values, the venous return, should be equal to cardiac output. Okay. All right. So all this blood is essentially moving through the cardiovascular system, right? And if we measure the volume per minute, leaving the heart, right or left, entering the heart, right or left, those values are going to be the same. Okay. Does anybody have a problem with that concept? Okay. So we can say if we've got a typical cardiac output is about 5 liters per minute. This is at rest. Okay, so most of us here, within reason, we're going to have a resting cardiac output of about 5 liters per minute. Okay, that's the left side, and it's going to travel through the circulatory system, and 5 liters per minute are going to be pushed through and coming into the other side of the heart at the same rate. Okay. And then 5 liters per minute leaves the right side, and 5 liters will come back to the left atrium. Okay? You okay with this? Okay. So, we understand that stroke volume and heart rate contribute to cardiac output. Okay? So, we're going to take a look here at some of the things that affect the heart. And mostly what we're going to be talking about is the autonomic nervous system. Okay? So you're going to have the sympathetic nervous system, which is represented here in green, and then you'll have the parasympathetic, which is represented in purple. Okay? And if you look just from the top, we can see that the parasympathetic nervous system is going to innervate and supply nervous signals to the nodal cells, AV node, SA node, right? And the sympathetic nervous system does the same, right? In green here, the sympathetic nervous system is going to send signals to the SA node and AV node, right? So they sort of mirror each other in this regard. One area where they're a little bit different is when we're talking about the actual muscle cell, the contractile cells themselves. What looks different here? What this diagram is pointing out. Between sympathetic and parasympathetic, what looks different? Yeah.
Doesn't the sympathetic also innervate the contractile cells or the parasympathetic doesn't?
Yeah. So the way it's shown here is you can see in green here the sympathetic nervous system is going to the ventricular. What do they call it? Myocardium. All right. So myocardium representing the contractile cells. Okay. There isn't much to speak of any innervation of contractile cells in the atrium by the sympathetic nervous system. Okay. If you look at the parasympathetic nervous system, it has an innervation to the atria, but not so much to the ventricles. And even the innervation to the atria is rather small. Okay, so there's not a lot of it. So we can moderate contractility with the autonomic nervous system, but if we're talking about what's sort of the biggest effect, or the most observable effects we can see in normal physiology and pathologies that result, we sort of recognize that there is a sympathetic control of the ventricles contraction strength, okay? And that's sort of where we leave it. Okay, we could But, you know, to be complete and to be thorough here, there is a little bit of iteration to the atria, okay, by the parasympathetic nervous system. So mostly what we're going to talk about is the chronotropic effects. Okay, so chronotropic effects are those that affect timing, chronos, right? So these are the, this is primarily the SA node. Okay. And we have ionotropic effects, which are effects on contractility. Okay, and this is mostly the ventricles. Okay. And the effect of this is to change stroke volume. Okay, so if you can change the contractility, you change the stroke volume. That is how strongly the ventricles are contracting. Okay. There's another effect here that we're not going to be spending much time on, but I want you to be familiar with what it is. Dromotropic effects. These are effects on the velocity of conduction. That is how quickly action potentials can move through the heart. Okay, so we learned about that whole pattern, right? From SA node, AV node, bundle branches, all that kind of stuff, right? So we have some neurons from the sympathetic nervous system in particular that will innervate those pathways and will have the effect of speeding them up. Okay? So these are called dromotropic effects, things that affect the conduction velocity. What does this mean? Well, when your heart is really under sympathetic control very heavily, the amount of time between when the atria contract and the ventricles contract will get a little bit shorter, right? They'll be a little bit closer together. So that's the major effect. Okay, so we're not going to spend much time on that because it's not nearly as important as these other things. Okay. One thing I want to be clear about is that there's something that these nervous connections don't do. They don't exactly tell the cells when to contract, right? Who tells these cells when to contract? Not neurons. If we cut your heart out of your body and held it up for everyone to see, it'd be cool. It would beat, right? Why is it beating?
Pacemaker.
Yes, the SA node. Okay, so the SA node, or cells acting through the action potential generated by the SA node, are the ones are stimulating contractions in the beat of the heart. Okay, so these neurons are only affecting the rate at which the SA node produces its action potentials, or they're affecting how strongly the chambers contract. Okay, they do not affect the timing of contraction directly. Okay, only in an indirect way by working through SA node. Okay, so those are the autonomic nerves. The other thing to keep in mind is, you remember way back when we learned about there are two neurons in a chain in autonomic systems, right? What do we call those? Preganglionic and postganglionic cells. Okay, the postganglionic cells are the ones that make contact with the effectors throughout the body. Okay, so if it's the heart, or the bronchi, or it's your kidneys, whatever. Okay, those are the effector cells. And we were talking about the postganglionic neurons, the ones that are forming synapses with those cells. Right? They have a very stereotyped release of neurotransmitter. Sympathetic nervous system will release norepinephrine to its target cells. Okay? And the parasympathetic nervous system will release acetylcholine to its target cells. Okay? All right, we call the receptors by different names, but the category of cells that respond to sympathetic nervous system neurons are called adrenergic receptors, and the one we're concerned with here in the heart is the beta-1 receptor. Okay, so you see that down there, beta-1. This is the receptor we're going to be talking about. This is a G protein coupled receptor. And the parasympathetic nervous system signals to cells that have on their surface cholinergic receptors. Okay, there's two types or two categories of cholinergic receptors. We learned about this once long ago. You remember what they were? Ever heard of nicotinic receptors? Raise your hand if you've ever heard of a nicotinic receptor. Okay, so somewhere deep in your brain, good. And the other is muscarinic receptor. So these are two categories. Okay, nicotinic you find on skeletal muscles. Okay. Muscarinic you find on targets of the parasympathetic nervous system. Okay, so this is what we're dealing with. We're talking about muscarinic receptors today, and we're going to be talking about adrenergic receptors. Okay, so adrenergic. Whoa. Adrenergic. Okay, the muscarinic receptor we care about today is called M2, and the adrenergic receptor we care about today is A1. Okay. So when we go throughout the rest of the course, we're going to encounter some other receptors, and there are going to be adrenergic receptors, and they're going to be muscarinic receptors. Okay. They're both G-coupled, G-protein coupled receptors. Okay, so how they work is very, very similar. Okay, so we'll run through this as a way to understand what's happening inside of our cells. Okay. Last time we spent a little bit of time talking about how there's all these different channels, right? And part of the benefit of having so many channels, despite the problem of having to remember them, is that pharmaceuticals can affect different types of channels, right? Calcium channels and so on. So the same is true here of these receptors. There are pharmaceuticals very, very commonly used that will affect, let's say, beta-1 receptors, but they will completely leave alone beta-2 receptors. Okay? So knowing where these things are and how they function is an important part of understanding how drugs are used, okay, clinically. All right, so we've got a neuron here, purple, right? We have a release of norepinephrine. A bunch of norepinephrine molecules are released. Right, and here we have a nodal cell, or a pacemaker cell. Okay. Got the beta-1 receptor in the cell membrane. Okay, and what happens is that our G-coupled, I should say, the G-protein coupled receptor has something it's bound to, or let's say coupled to. Anybody know what it is? A G-protein. So G-protein coupled receptors are connected to G-proteins, and G-proteins have three parts. That's what's shown here: alpha, beta, and gamma. And what will happen as a result of the binding is that we'll release that alpha subunit. Okay, the alpha subunit will pick up a molecule of, who remembers? GTP. Okay, so it'll pick up GTP, which will allow it to bind to an enzyme. Okay, this enzyme is adenylate cyclase. Okay, AC. Cyclase. Okay, so we have an extracellular signal resulting in an intracellular effect. Okay, that adenylate cyclase is an enzyme which will take ATP and convert it to cyclic AMP. Okay, that's where the cyclase part of this enzyme comes in. ACE means it's an enzyme, and it's converting a molecule to a cyclical form. Okay, so we get cyclic AMP. Cyclic AMP is our what? What do we call that? How do we classify it? It's a second messenger, right? So that tells us it has this important role in the cell, that it is a signal that's going to carry on to many other targets throughout the cell. The effects of our signal. Okay, our first messenger is norepinephrine. Okay, so we make cyclic AMP. Do we make one molecule, or how does this work? We make lots of this. Okay, so this is an amplification effect. This enzyme is active for a period of time. Okay. All right, so we make lots of cyclic AMP. Cyclic AMP goes and it activates a kinase in our cell. And what does it mean to be a kinase? It's an enzyme that does what? Phosphorylates things. Okay. The term kinase literally means an enzyme that activates, right? But we understand kinases to be proteins, enzymes, that have the role of adding phosphate groups, or in other words, phosphorylating other proteins. Okay, so when we phosphorylate another protein, we've activated it. Okay, so PKA, or protein kinase A, is an enzyme that functions as a kinase, goes around and it activates a bunch of things in the cell. Primary target here in our nodal cell is that it's going to go and it's going to phosphorylate our calcium channels. Okay, remember our nodal cells have these T-type calcium channels, and what's going to happen as a result of that, what happens, we increase permeability to calcium. Okay. So that's a long way to go to get there, right? And it's complicated, right? So we have a first messenger, what we call the second messenger because it's sort of the first step of amplification. Okay. And then we have an effect on our cell. Okay. So as a result of norepinephrine signaling to this cell, the effect is that calcium can more freely flow through the cell membrane. One other thing to know here, cyclic AMP will act on not only protein kinase A, but has another target as well. What do you suppose it is? Hint, there's a 2 next to it. HCN channel. Okay. Who remembers what HCN stands for? Hyperpolarization activated. Or hyperpolarization, what was looking back at that? What's the CN?
Cyclic nucleotide.
Cyclic nucleotide, okay. So who's our cyclic nucleotide in this image? Yeah, it's a cyclic AMP, okay. So we have a channel that is going to be activated by hyperpolarization, but it's also going to be activated by cyclic AMP. And its activation in this regard with cyclic AMP is that cyclic AMP actually binds to it and increases permeability. Okay, so the more cyclic AMP, the more permeable our HCN channel. Okay, so both the sodium and the calcium channels become more permeable in our pacemaker cell. Okay. So if you could, on your own, pull everywhere and tap the screen or click the screen at the appropriate place to tell me how norepinephrine is going to change our nodal cell action potential. Which part of that action potential is going to be affected? Is it active? Who needs more time? Okay. Who needs more time? Okay, good. All right. All right. So, most of you, I would guess, most of you sort of prefer the upswing than the downswing. We've got a lot of difference here. Somebody tell me if you have a strong opinion about this. Where do you think this is? What's going to be affected by our norepinephrine signal? Yeah. What do you think? The T-type, where does the T-type channel, where is that important in the cycle of the action potential?
It's after the hyperpolarization.
Yeah, okay, so it's after the hyperpolarization. And somebody want to add to that? What else besides our T-type channel? HCN? Yeah.
HCN.
Yeah, so where are both of these things active? The lower part, what do we call that? The pacemaker potential. Okay, so the pacemaker potential is this portion that's right in here. Okay, this is an odd placement, by the way, Full County. Yeah, so this slope where it's going from its point of hyperpolarization, it's climbing, right? And the reason that it's climbing upward is that we have sodium and calcium coming into the nodal cell. Remember that? And that is happening in our nodal cells, and it's moving us towards the threshold. Okay, and what happens when we get to the threshold? Something else opens up, right? What is that? We have the big depolarization phase. What's responsible for that?
Voltage gated sodium channel.
Voltage gated sodium. It's the L-type calcium channel, right? So remember in this one we've got the T-type, which is along with the HCN channel responsible for that pacemaker potential here. Okay, and then the L-type channel opens up here, and then potassium brings us back down. Okay, so if we take an understanding of our pacemaker potential being the result of these two channels opening. And in reality, the HCN channel opens and then the T-type, but they're both responsible for this slow rise up to threshold. Okay, and so the effect that we see when we open up these channels is that with more positive ions coming in, we get to threshold more quickly. Does that make sense? Okay, so positive ions coming in means we're depolarizing the cell membrane, and then depolarizations lead up to threshold. Okay, and so what's happening here is in the purple line, we're getting up to threshold in less time, right? Because we have more ions coming in per unit time. Right? Everybody see that? We don't really have an effect on the depolarization phase or the repolarization phase, but that pacemaker potential is what's affected by the action of norepinephrine. Okay. So with each of these taking a little less time, what happens is that instead of your action potentials occurring on a infrequent basis, they happen more frequently. Right? So they're a little bit, each action potential is a little bit closer together in time due to the fact that the pacemaker potential is shorter. That make sense? We good? Does it bring up any questions? Okay. All right, now, norepinephrine is not the only thing that can stimulate this receptor. Okay, so we have a similar molecule called epinephrine. Okay, norepinephrine is usually abbreviated NE, epinephrine is E. Anybody know where epinephrine comes from? The adrenal medulla. Remember the adrenal gland sits on top of the kidney. You've got a cortex on the outside and the inner part called the adrenal medulla. Okay. When would this adrenal medulla be releasing epinephrine? Under what circumstances? I know you didn't read about it today, but remember back to whenever? Yeah, the fight or flight response. Remember that? Now you remember. Okay, good. The fight or flight response. So this is the whole body's systemic response to sort of a short-term stress, right? Running away from the dinosaur or something, right? You're suddenly alert and your body is prepared for action. Okay. So norepinephrine can act through the sympathetic nervous system and be stimulating the heart to go faster for reasons like maybe your blood pressure drops and you need to raise heart rate to make cardiac output. Right? So to respond to sort of normal reflex things. But when it comes to the fight or flight response, we release epinephrine. Okay, so there's sort of two different reasons. One, norepinephrine is more used in reflexes. Epinephrine is used in the whole body response to stress. Okay, so it's a hormone, travels throughout all the circulatory system and affects not just the heart, but everything. But the easy thing to remember here is that both norepinephrine and epinephrine will bind to the same receptor. Okay, so epinephrine can bind to the same receptor, and it can either replace the activity of norepinephrine or augment it, work together with it. But in any respect, epinephrine will stimulate the very same pathway. Okay, so this G protein coupled receptor pathway we've learned can be stimulated by two different hormones. Why have two different signals? Different circumstances. Okay. The neuron is participating in reflexes. Epinephrine is there when we have a fight or flight response. There's a third hormone called thyroid hormone. It comes from the thyroid. You guys are on today. All right. There's something, this one works a little bit different, but this is an important principle to know, not just for the heart, but for other parts of the body. When is thyroid hormone released, or why is thyroid hormone released? What is it doing generally? Not to the heart, but just generally in the body. Do you have a sense of that? About sugar. Yeah, directly. It's sort of the major metabolic hormone. It raises metabolic activity. Okay. So when you go out outside and it's cold out, and let's say you've got a job you're working outside, right? In order to generate more body heat, your thyroid hormones can grow. Okay. And people have disorders of the thyroid gland. If you've got too much of it, you lose your body mass because you're consuming too much energy. You're very hot and very skinny, right? Or if you have hypothyroid, not enough thyroid hormone, your metabolism is low and you tend to gain weight and have a lower body temperature. So it's a hormone that is responsible for sort of revving up metabolism, okay? And that has a lot of effects with regard to oxygen and body mass and all these things. So what does it do in the heart? Well, thyroid hormone is one of these that acts like a steroid hormone. In other words, there's no extracellular or plasma membrane receptor for it. It passes right through the cell membrane, and it binds an intracellular receptor. Okay? And it has a receptor that's called the thyroid hormone receptor. Easy enough, right? That's what THR is. And what they do is that they will together bind to DNA, right? And they will transcribe genes. Okay? And these are genes that are concerned with metabolism. Okay? But in the heart, one of the things that it does is it transcribes beta-1 receptors. Okay? So what happens when you activate transcription here is we're going to get a bunch of mRNA. Okay? More green cones. mRNA gets turned into, everybody says, proteins, right? ER and Golgi, all that kind of stuff, right? Biology 101. All right, okay. And so the Golgi sends out these little vesicles with the proteins in them, and they fuse with the cell membrane, and before you know it, you've got all kinds of receptors added to the cell membrane. Okay? So this works a little bit different. It's a little slower, right? It's not immediately happening like with the second messenger system. But what happens is we make more receptors. Okay? So let's walk ourselves through this. With more receptors, what does that do to our sensitivity to epinephrine or norepinephrine? Increases sensitivity. Okay? So therefore, even with the same amount of norepinephrine, we're going to get more signal. That make sense? So three hormones, norepinephrine and epinephrine, working through the G protein coupled receptor pathway, directly stimulating the shortening of the placenta potential. Okay. And the thyroid hormone acting longer term, increasing sensitivity to those other hormones by adding more receptors. A principle you should know is that thyroid hormone has this effect on all adrenergic receptors throughout the body. Okay, so not just beta 1, but all these other ones. Okay? And this makes sense, right? If you're going to be elevating metabolic activity, we're going to also want to affect the heart. The heart's going to have to work harder to move blood more quickly to deliver more oxygen. Okay? Because we're consuming it. So that makes sense, right? Okay. Questions about those three hormones? Who's having no cell biology for today? Answer this question, then we'll move on. Talk with somebody close to you to get a better answer on average. So under which of these conditions would you not expect a faster heart rate? Okay, fight or flight response, working in a cold environment, injection with epinephrine, which is what we do with an EpiPen for different reasons other than the heart, but and then relaxing on the beach on a hot day.
Like if you let's do it fix fight or flight, we know would increase. Working in a cold environment instead would increase. Because you're gonna get more body injection with epinephrine. Epinephrine does increase the heart. Because relaxing on a beach on a hot day, even though you're sweating, like you're trying to
Does temperature play a factor in heart rate, right? So is it increase in temperature equals increase in heart rate? So would it be a better
But I feel like if you're trying to cool your body down, your heart rate's lower. Because when you're trying to cool your body down, your heart rate drops.
Okay, the question's too easy, right?
Is it?
Well, you've answered so quickly. Okay. It's too obvious, right? No, this is hard. This is a hard question. Yeah. My cursor is invisible. All right. Let's walk through these real quickly. What's happening in the fight or flight response? What hormone is going to be released?
Epinephrine.
Epinephrine, right? What's epinephrine going to do to you?
Increase your heart rate.
Increase your heart rate. How? By shortening the length of time your pacemaker potential takes to get up to the threshold, right? Working in a cold environment, what's going to happen there?
Increase.
Increased production of
Thyroid hormone.
thyroid hormone, right? Thyroid hormone responds to maintain homeostatic levels of, or homeostasis of your body temperature. That's one of the things it does, right? So in a cold environment, it's going to raise metabolic rate in order to keep your body warm enough. But on the heart, what's it going to do to those cells? Increase sensitivity, increase the number of beta 1 receptors. Okay. Epinephrine, this is part of the fight or flight